we still need a lot of knowledge and just try to understand what is the what is the basic nutrition of a dog and cat. And then I go to the ecology of the animals and then try to understand how far are we away from that specific trait of the nutrition that we observe in nature that is changed in how we are feeding our dogs and cats in home. And then you come into all different types of questions.
Good morning, good afternoon, and good evening. I'm your host today, Dr. Kate Shoveler, and I'm here with Dr. Guido Bosch from Wageningen University and Research. Before we get started, Guido, why don't you tell everybody a little bit about you in case they're not familiar with you and your work.
Okay, thanks for the introduction, Kate. I'm Guido Bosch. I'm working here in Wageningen, the Netherlands, a tiny country in Europe. I'm specialized in dog and cat nutrition. I did my PhD over here for the effects of nutritional behavior in dogs. So I like to combine nutrition and behavior in my research. I also have an interest in ecology of animals, methodologies, feed processing technology. So it's quite broad in terms of topics and also animal species. So I also go beyond dogs and cats. So zoo animals and insects are also on my plate, so to say.
So a very unique comparative nutritionist, because I think the majority of us who do work with multiple species tend to stay in the mammalian kingdom, and you definitely have branched out a little bit into other kingdoms. So congratulations for that, because that's not easy to do. I thought too, and mainly because I've spent the last year with you. So for the listeners out there, I was on sabbatical at Wageningen with Guido and his fellow animal nutritionists there. And it's also an outstanding university in terms of what's available to do research. So do you want to describe a little bit too what's available to the pet food industry and to the ingredient industry when we think about contacting you to maybe sponsor a graduate student's program or to do research for that matter? What's available to you and your colleagues?
Okay, so on campus we have different types of research facilities. So very specific for pet nutrition, we have cats over here, 34 cats. And the cats are trained for behavior research, but also nutrition research, so they are well behaved. Not stressed, of course, they are well habituated to all the procedures. We can do digestibility work, more the standard type of work, urine collections, but we also look into food motivation, for example, feeding patterns, and have developed some equipment to measure these kind of things. We also have laboratories to do just standard proximate analysis, but also a little bit further, so fatty acid profiles, amino acid profiles, minerals, in vitro digestion simulation we can do. And also fermentation in vitro is something that we can do on a quite large scale. So many different types of treatments can be evaluated at the same time. And other than that, we also have other groups on campus that have specific expertise and also laboratories. So if you want to do something that is more on the chemistry side, then we collaborate with the Laboratory of Food Chemistry or on microbiology or entomology. So we have lots of facilities around us and people that are open for collaboration.
Yeah, and it was an extremely lively campus. I think that you're quite fortunate to be there. The programs and the research are bar none some of the best in the world. And so it was a lot of fun to observe and be able to take parts in some aspects of that, you being more familiar with what I was involved in than the listeners. So I think that today, I thought it would be really interesting for the listeners because it's something that we're very interested in, both of us personally as scientists, but your most recent research where you're kind of pairing up moving digestibility studies to in-home, then additionally thinking about how to complement those with in vitro. And of course, I think that I'm not going to be able to have this discussion without kind of also talking about how the entire group, animal nutrition group at Wageningen, is really interested in vitro, and how maybe our species models that we use for digestibility are or can be applicable in some situations for dogs and cats. So why don't we start a little bit with the in-home movement that you made, where you're looking at taking these classical laboratory digestibilities and bringing them in-home. Would you like to describe how you got that started?
Yes. Actually, Wouter Hendricks, the professor over here, started this, I think about 10 years ago, a little bit more, because he had an interest in setting up these types of methodologies, and he started with some students, did some first work that was also presented in one of the conferences. And over time, he also got into contact with the industry, with companies that also have an interest in moving away from the kennels, which dogs have or are being used to do the digestibility type of work. And at a certain moment, there came an opportunity to set up a PhD project. So there were more companies willing to invest money to develop the methodology. So together with the University Fund of Wageningen, we set up a project, attracted more companies. So we had a pool of 10, 12 companies that all chipped in. Then we could hire Guido Bosch, who is now finishing up her PhD thesis, and started to work. And so we are doing both digestibility type of work as well as adaptability, and then in dogs and in cats.
Yeah, I think that this is, first of all, I commend you for getting multiple stakeholders together, because this is really a methodology that the entire industry needs. And one thing that we've already discussed with other people on the podcast is really the need for academia and industry to partner to solve solutions. And Wageningen has done that really well. So when you were putting together the effort to support that work, was that difficult, or was it quite easy? Everybody was mutually interested in in-home digestibility.
It was quite okay. I think there were some founding fathers, so some companies that were very enthusiastic and really were pushing it, and they were really also helpful in convincing other companies to help out. So they have a strong network among each other, of course, and they could also convince the other pet food companies to contribute because it's a mutual interest for everybody. And as such, you also need to work together to get such type of methodology developed, and everybody can benefit from it. So actually the companies amongst themselves did most of the work. We also had our own network, but I think in the end, the companies were most convincing to their colleagues, and that really helped to get all the money in.
Yeah, that's a really interesting approach, and great to see that collaboration as well within the industry. There is a little bit of truth to that. If an industry takes a hit, so as an example, if kibble takes a hit, it can affect all kibble. So it's nice to see everybody in different parts of the sector working together. So Eveline largely looked at dog, correct?
Dogs and cats, both.
And she did do cats. I don't think I'm familiar with that work. Was it a little bit, I'm going to assume that the cat was a bit more difficult than the dog. And just curious, because one of the things that's really hard is that owners tend to be with their dogs when they defecate. So you can get your whole sample usually quite effectively, and it's fresh. How did she translate that into cat and make sure she was getting fresh fecal samples, or relatively fresh fecal samples?
Yeah, there was a challenge, and I think the recruitment was also a bit more difficult, because with the dog owners, they are really in control in terms of what the dogs are eating, and when they are probably defecating, where they defecate, and so on. They can help out a little bit, and it is a little bit more easy to collect the feces. Although she also has some issues with the owners that were not able to go into the bushes and try to find the feces of the dog. But apart from that, we had to contact cat owners that were having the cats only indoors, because when they go outdoors, they might also eat rodents or birds or whatsoever. So you cannot really do an accurate digestibility measurement. So that already narrowed down the population of cat owners. Sometimes cats are also a bit difficult in eating different types of food. So it's a little bit more tricky to get the recruitment going on for the cat work. The feces collection is more easy. You don't have to look for it. You know where they are going to defecate. So that's a bit more simple. But the freshness of the feces is not always guaranteed. And it's also not always needed for a digestibility measurement. So only if you want to do like fecal fermentation products, then you need fresh feces, but it's just like a regular digestibility measurement in which you want to evaluate nitrogen digestibility. There's not too much nitrogen going into the environment.
Yeah. So maybe for the listeners too, most of them are in the pet food industry, but this is a good opportunity to kind of delineate the difference between when you're collecting a fecal sample to do a simple digestibility versus a fecal sample to look at the bacteria that resides in the feces, the products of fermentation. Maybe for the listeners, describe what the improvement in your understanding of the effects of the diet would be as you add different sample collections or analytics to your fecal samples.
I'm not sure if I really understand your question.
Yeah, because you mentioned, so I was asking about digestibility, and indeed, you said that for having a fresh fecal sample to do something like nitrogen digestibility isn't an issue, but might be an issue if you were looking at products of fermentation or you were interested in the microbiome, for example. Maybe for the listeners, and it takes a lot more effort. So the effort it would take to get that kind of sample, but what it might be able to tell companies about their diets if they invest the money in supporting the staff to collect those or the technologies to collect those, and then being able to do additional analytics.
Yeah. Okay. Thank you for clarifying. Yeah, that's indeed interesting, because if you have fresh fecal samples, then you can look into these microbiota or the metabolites of the microbiota that would say something about what is ongoing in the intestinal tract, in the large intestine. So it might say something about the protein quality, for example. If you have lower digestibility of proteins, you might have more protein fermentation going on in the large intestine, which will give you a different fermentation profile in terms of metabolites, but also in microbiota. You might also have the possibility to collect some proteins that are secreted in the intestinal tract that are coming from the immune system, so something that is related to gut health. And in essence, we don't really understand at this moment how we should relate the microbiota, so specific species that are maybe very much present or are reduced in numbers because of a dietary treatment, and also the fermentation profiles to actually gut health. So what is very detrimental or maybe beneficial for gut health on the short term or long term, at this moment we don't have a very good idea. So this is still a challenge. And I forgot to mention with the cats and the collection of non-fresh feces, it will be also difficult, of course, to evaluate the fecal consistency, which is more easy when you have the dogs. And the fecal consistency is also often a parameter that companies want to have. But for the cats, it is difficult to guarantee that.
Yeah, logistically, and I'm just curious, and for those listeners that haven't done digestibilities with cats, there's another thing that we ran into, which was we had our first colony of cats were actually all Humane Society cats, and we conditioned them and did some litter experiments on them. But we couldn't adapt them to a AstroTurf to urinate and defecate on. So we used non-absorbent litter. But then to do fecal analysis, you have to pick out every single piece of litter off of the piece of feces. So it's for people who are thinking about asking about research, some of this stuff that we have technicians and students do is extraordinarily laborious in nature and slightly unpleasant. So did you have to do the same thing? Did you make Abeline pick out all that litter from all those species before she ran them?
Yeah, in parts. So what could be removed was removed. And it was also very laborious. And we actually also did a compliance manuscript. We're now working on it. And that includes, of course, compliance to the dietary treatment, not giving too many treats, and also the understanding of what confounding factors might do with the estimate of the digestibility. That also includes the litter that might be attached to the fecal matter.
Oh, interesting.
And yeah, so in essence, if you have a dry matter digestibility and you have lots of litter attached to your feces, then you cannot really do an accurate measurement. But if the litter is devoid of any nitrogen, you can do an accurate nitrogen digestibility estimate. And we also use the marker methods. So maybe that's also an important detail to mention here. But yeah, Evelien also tried to quantify what is the amount of litter that is attached to the fecal matter.
You are the meanest advisor ever.
Yeah, we also had lots of students that were also helping out. So we tried to maybe spread the burden a bit.
That's an interesting measure, though. Yeah, those are so dog and cat are extraordinarily different. And I think what's really interesting is also that you're looking at variables that might affect digestibility. And of course, in a laboratory setting, we tend to, especially animal scientists, tend to overly control the environment and all the experimental parameters. What are you learning about the variables that can affect digestibility that are animal specific rather than diet specific?
Yeah, obviously, if you look at dogs, then body size is an important factor. Activity can be an important factor because it's also affecting food intake, and that might relate then to passage rate and defecation frequency and so on. Yeah, so these types of factors are quite important. And I think if you would do a trial within a kennel, I think it's also very important to have these facilities if you want to evaluate specific ingredients or specific research questions that require a certain level of control in your study. If you are a bit more close to launching a product to market, then it makes more sense to evaluate maybe in the target population. So you might have different, or you need different facilities for your specific question that you have as a company.
Yeah, I think we definitely see, and you're doing a great job kind of dovetailing into some work that we mutually love, which is really, trying to understand the digestibility of ingredients and how those ingredients then relate to digestibility of the final diet. So let's talk a little bit about those digestibility measures, and because you mentioned, really closer to market, and I totally agree. But what about when you're for example, what would you suggest to companies who have an ingredient supply shortage and have to make a quick similar ingredient change? Let's say there's very few digestibility measures on that new ingredient. So maybe a great way to kind of into another realm that you're involved in is they want to start using insect meal. They have a shortage on let's throw out some fish meal and chicken meal in there. and I think what we see is a lot of companies do a swap. But in this case, where you have limited amounts of information on the digestibility of black soldier fly larvae, although I will admit that I think it's growing quite rapidly, because you do need multiple studies to kind of contribute to that body of knowledge. But what do they do there? So if somebody wanted to do fast, rapid development to market with black soldier fly larvae, do they run an in-home? Do they run a control? Do they run anything at all? What would your recommendations be to the industry?
If you want to understand your new ingredient, then just plain chemical analysis, of course, is helpful. And then you can complement that with in vitro digestion, just to compare the in vitro digestibility with the reference ingredient that you have. Then you can have a good idea of the quality of the new ingredients that you want to incorporate in your diet. You have to be sure that it's safe, of course. If there are any issues around amino acid composition that you know of, then you might also run an amino acid digestibility analysis with the in vitro method that you have. That's also an option. It's not that common to do, but you can do it. And if you have more time and facilities are available, of course, you can then go into a test with a kennel or a cattery to evaluate your product, but it also takes takes more time to organize, to find a lab and so on. But in essence, you can do quite some first steps on a short notice using laboratory methods.
Yeah, so that really kind of dovetails nicely into in vitro methods, and I got exposed to a lot of the nutritional physiologists at Wageningen using different in vitro methods. So, and across multiple species. So maybe talk about and you also differentiated, you talked about amino acid digestibility separate from other digestibilities. So let's start there, because you and I are really passionate about protein. If they're really concerned about amino acid digestibility, what in vitro approach do you recommend to companies to take a quick look at ileal digestibility of the amino acids?
Yeah, there is no validated method. So each method that is out there and published is not validated. So you always are a bit unsure to what extent your results are valid. But you can use the in vitro methods that are described in the literature to evaluate your ingredients or foods, and also to rank them, for example. And so you have a systematic error there. We have a couple of methods that were developed for pigs that we are also using here in Wageningen, and that were further advanced or tailored towards the digestive physiology of dogs in Spain. So that's described by Marta Rivera. So we have that over here. And then they changed the pH, for example, of the gastric phase, the incubation time, according to the digestive physiology of the dog. So that is something that we are doing. But there are also more advanced methods that are more dynamic, for example, with the TNO model, where you have really peristaltic movements in the system. So it's way more dynamic. The collection of the digested part or the undigested part is different. And there are also some drawbacks in how to, in the end, differentiate between the digestible fraction and the undigestible fraction, where you're going to do, for example, filtration versus centrifugation. So there are some issues around that.
Yeah, we attempted in some swine experiments to use the tip one, and I think maybe I would add that it is an incredibly finicky in vitro method that requires a tremendous amount of oversight, an extraordinarily knowledgeable technician, because you do, like you said, it's dynamic. You in essence have a ex vivo in vitro system, and it needs to be curated pretty closely, or you have issues and need to do reruns quickly. So I think that those kinks really need to be fixed in that too, because I remember being on the other side of, so not in academia, but in industry, it had to be 15 years ago, at the American Society of Nutrition. They were having a really big symposia, really talking about DIAS methods for human nutrition, which we apply to dog and cat. But all the industry people were saying, you can't give me a 2-year timeline to screen an ingredient. I need a 2-week timeline to screen an ingredient. We have to make decisions really, really quickly. So I think for the industry, that kind of culmination of the in vitro methods that become validated, I think will be tremendously beneficial because of just the timelines and the cost of doing in vivo.
May I add to that? Yeah. The FAO is running a program. It's called Proteos. And in that program, they are evaluating protein sources for human nutrition. And they are using the pig as a model. So they have ileal digestibility data for different protein sources. And in that whole project, they are also validating the in vitro infogest model that simulates human digestive physiology, and they try to optimize it so it is validated together with human data. So they have human ileal digestibility data, pig ileal digestibility data, and in vitro digestibility data. So they then can correlate all these 3 ways of measurement and validate and optimize also the in vitro method. And I think that would be also very nice to understand how these methods will look like in the future, because the digestive physiology of a dog and maybe also of a cat will not be that different from a pig and a human. It will not be day and night different. So actually, I think that we can maybe make a step in improvement of these in vitro methods to get more close to real data in the upcoming years. So that will be interesting, I think, to follow.
Yeah, and you bring up one really good point too that I think you and I think a lot about, but we get caught a lot in pet nutrition where there's a need for species-specific data. And I think that there's some people who get a bit distracted and think that that means that in every single thing that we do, we need species-specific data. And I think that when it comes to digestion and absorption, we see less differences amongst species. And that's the important thing when we're looking at a digestibility model and cross-application of that data versus looking at how an animal peripherally metabolizes a nutrient, or the difference between when in the day or when in the week or when in the year they metabolize more or less. And I know that you're really passionate about kind of you probably think a little bit too, well, I know you do a little bit, about the implications of kind of those diurnal rhythms in dogs versus, what are cats? Are they crepuscular in nature? That's certainly what the indication is, but they're so highly adaptive. So just because we like to branch off and talk about things, maybe talk a little bit about how you would overlay the nutritional ecology of the dog and cat separately from applying digestibility to things like to species like humans or agricultural animals, because dogs and cats are closer to humans in the fact that they're free living, so they're a little bit more in control, yet they're in less control usually of the food items they eat versus humans who are in control of the food items they choose to eat. So what are your thoughts on that and how we think about that digestibility data in these two animals that share our lives but are different seasonally on daily rhythms, and frankly on their ability to communicate those to us?
It's quite complex. So if you look into digestive physiology, I think any organism tries to get most out of the diet that is being eaten. So also for the dogs and the cats, they have their limits in terms of how they can digest certain parts of the diet, but they can adapt, as you also mentioned. Like the cats, they also change their activity according to when food is available, for example, or when partners are available, and so on. They can do time sharing over a certain neighborhood, and so on. It's really nice what they are doing. But also in the intestinal tract, they can adapt quite well. And so if you look into the feeding ecology of a cat, it's a carnivore, but we already know that they can digest starch quite well. So the starch digestibility is of the total tract is over 90%. And I know that Darwin cited an observation that in one of his books about the domestication of animals, and he described that somebody observed that cats that were fed table scraps have a small intestine 1/3 longer and wider than the cats that are living in the woods. So that also shows that the cats can have quite some severe changes in the intestinal tract just to accommodate a certain food composition that is a bit unnatural, but still they can cope with it. And if they are being overloaded with starch, then they would get diarrhea. So we also have clear understanding what the limits are, I think, what we can feed the animals in terms of digestion. So we are not talking about the physiology and metabolism, also long-term health implications, but in terms of digestion, we know that they can cope quite well with quite extreme types of diets.
Yeah. So why do you think? Because I always think it's quite interesting that if I walk into a pet store and I'm looking for food, and I do this quite often. I don't know if everyone else does, but I like to see what they know and maybe what they don't know or what they believe. And so I've often tested that I'm going to switch my cat's diet or my dog's diet. And I almost always do a kibble exchange. And no one ever asks me anything about my dog or cat, but they're like, well, their GI tracts are really sensitive. So, take one food and change it really slowly, or your dog's going to get diarrhea or your cat's going to get diarrhea. But is that really the case? I maybe talk about kibble to kibble, kibble to wet, kibble to raw, and how you would do those if you were a pet owner or you were advising pet owners to switch to a different format.
Yeah, so in our cat facility, we also switch diets, and we also don't observe issues with the rapid switch from one diet to another, so from kibble to kibble, but also not from wet to kibble, for example. So wet being lower in starch and kibble being higher in starch content. And in one of the studies of Evelyn, so the dog digestibility study, there was also a subset of dog owners that were feeding their dogs raw meat type of food, and they were switching to the dry kibble to evaluate the digestibility. And we also did not observe any issues with changing from that diet to the kibbles, and also we did not see strange effects in the digestibility values of these dogs. So apparently they were well capable, at least these dogs were well capable of coping with the switch and also digesting it.
Yeah, so one thing I always wonder about this is, do you think that by using colony dogs and cats who we do tend to do very thoughtful but often changes in diet, you and I are dealing with changes in personnel and constantly training new and exiting others. So it's there's constant change with colony animals. I always wonder about and I have thoughts given maybe some hermetic challenges that the animals would have with a constantly changing diet. Do you think, you have a cat? Do you feed him the same thing every day at exactly the same times? What's your method with Jack that you use in terms of feeding an approach that you could cross apply?
Yeah, Jack is being fed wet food and dry food. He's very food motivated, so we have to make sure that he's not overeating. So I really have to keep an eye on his body condition, and he gets 3 meals a day. In the morning, a bag, a little sachet with wet food. In the evening, when we are ready with dinner and cleaning up everything. So we are very routine oriented. So we have quite fixed routines in our household. And then in the evening, before we go to sleep, he also gets a bit of dry food.
That's to make sure he doesn't wake you up at 3 o'clock in the morning asking you for food, though. Yeah.
That is the idea. But still, he can be quite active in the night. I'm not sure if he's trying to get us out of the bed, but doesn't work.
I had an externally food motivated cat too, and when he was younger, he would do things like I remember once he ran up the bed and jumped on my chest and then ran out, and that was enough to get me up, and I got the hint that it was I was needed for the food provision that I was apparently a half an hour late on. But yeah, they can get so there's a lot of managing, and I know that people do this, a lot of owners do this, where they're just managing, trying to make sure that interaction with their dog or cat over these things are quite timed out. So let's maybe return here, because you mentioned something earlier, and you got me super fascinated about nutritional ecology. I think I was always like a little bit interested, and you've made me a lot more interested. You somehow have convinced me to look at different types of formats that cats would eat, from natural kind of prey items all the way to extruded and wet food. What about their place in the nutritional management of owned cats? Because you're very interested in natural prey and the behavior needed to consume natural prey, I think, is where you're focused. But how does that fit in? We've been talking about digestibility, and you've already said that the cats and dogs switch to different types and formats of food in terms of ensuring they get the most nutrients out of their food. We know that changing their food isn't an issue and could be of benefit for diet diversity. But what about these roles of, we took 2 hunters and we removed something that they spent a significant proportion of their time budget on in the wild? What are your thoughts about, first of all, missing that part, but then second of all, just the act of consuming those different types of foods?
Yeah, regarding the behavior, I think it's difficult to imagine how the dogs and the cats see their lives or the environment and experience their lives. And maybe some cats need to be challenged a little bit more, so they might end up in an apartment having a bit of boring life, but they sleep a lot and so on. There is research also ongoing, trying to understand if these animals can also have feelings like depression, if they are not being housed in a very enriched environment. But we also know that, for example, if you play with the cats, they might hunt less. So they have like an innate, or they want to do something. It's fulfilling for them to do hunting games, for example, and just it's in their nature, so to say.
Well, that's the reason why cats play well into adulthood, though, right? Is one of the conjectures is that they continue to play because it helps them kind of hone their hunting skills to an effect. So maybe this thing that we're talking about, where they're spending all this effort running around and with the zoomies and jumping on beds and trying to get us up, maybe that's all part of the activity of that time. So there has to be activity maybe around food.
And maybe it might also relate to the selection for tameness. So there you get a bit more, I call it these immature young type of animals that, so if you have a young animal, young animals really like to play because they are shaping their behavioral repertoires and that they need for later in life. And if you select for tameness, like we have done for dogs, if you look at the behavior of dogs, it's a bit disorganized. They are very playful, the adults, and that looks like wolf puppy type of behavior. So they retain that capacity. And people also say that humans retain these juvenile traits. It really helps them to stay creative, and they play a lot and so on. So we also domesticated ourselves a bit because of the benefit of being creative and just play around, be innovative, create new solutions to problems that we encounter and so on. That's also very, very interesting, I think. For the dogs and the cats, and when it comes to nutrition, I'm not really in favor of the raw type of foods, and I'm not against the dry foods or the wet foods. I think we still need a lot of knowledge and just try to understand what is the basic nutrition of a dog and cat, and then I go to the ecology of the animals and then try to understand how far are we away from that specific trait of the nutrition that we observe in nature that is changed in how we are feeding our dogs and cats in home. And then you come into all different types of questions like not only the nutrient profile or the physical structure, the stimulation of chewing, or the rate of gastric emptying, the importance of structure in stimulating intestinal motility, or all these types of things, the microbiota, fermentation profiles, what is normal to observe in feces, the metabolism, of course, and that is something that you will look into with the cats. So it's just like trying to advance our understanding of dog and cat nutrition just by combining some insights from cats and dogs that have more like a natural type of diet and see how it looks like.
Yeah, I think that, we look at it as we're focused on food, but then there's also the ability I do quite a bit of work in horses and I find that horses and cats and dogs, sometimes the technologies fly similarly, where you're seeing all these technologies that help you kind of manage that feeding experience with your animal, or in the case of cats, make things where they have to work harder for their food to kind of mimic that activity that has to be involved in obtaining the food. So it's not just the format of the food that we can look at, but the ways in which we deliver it and whatnot. But I think, too, your thoughts too on, you use the word normal. What is normal? And maybe how diverse normal might be among, especially, I think, the canine population in terms of diversity, maybe a little bit less in cats, but still quite a bit of variability, right? In your opinion?
Yeah, definitely. There's quite a lot of variation. So I'm not sure what the question is, but there's quite a lot of variation in how we are feeding our animals. And many of the animals, they just thrive and they become old in a healthy state. And so we are doing quite a good job, I think, in that sense.
Yeah, I think so too. No, my point is that because of the word normal, I think that some people are looking for us to say, okay, you tell me what's normal for all dogs and cats.
Yeah.
And we've already talked about how the dogs and cats are variable in themselves. And I think, maybe the other thing is to consider how variable the owners are. And how tapped in our dogs and cats are to their owners and their own. We talked about that at the beginning of the podcast. But I always talk too about people desperately want me to give recommendations. And, one of the things that I believe very passionately is that dogs and cats make our lives better. And I work to help further support that relationship as you do with knowledge acquisition, so we can continue to help people understand what dogs and cats need to be the best partners to us. In terms of breadths of products on the market, do you think that we're doing a good enough job with the breadth of products for the number of consumers and the number of pets?
No idea. There are so many different types of products, so it just, I think, mirrors the different values that owners have and beliefs that owners have. Because then you can only be successful on the market when there is like a consumer that really believes in the things that you are trying to sell and the concepts that you have applied in your product. So I think that is one of the things that is explaining why we have so many products, and our own nutrition and our understanding of what is beneficial, what is less healthy for us. There is also lots of developments and new trends, and that often translates just in the pet foods. It doesn't mean that these are also very much healthy for the dogs and the cats, but it will give some more breadth to the products that we have available in the pet shops.
Yeah. No, I completely agree with you too, and I think it is important to understand. I remember as a scientist when I went into industry, and I really wanted to kind of hammer on people about what was, what they could do better with their nutrition for their dogs and cats without asking any of those questions about their own belief systems and how much time they had to dedicate to this, how much money they had to dedicate to this, how they're balancing different parts of their lives with the ownership of pets too. So I think taking a human approach and understanding your consumer as well is super important. I'm glad that you pointed that out. So I could keep talking to you forever and ever, Guido. I will greatly miss having the opportunity to walk into an office and chat with you almost whenever I want. But we are going to have you back. I really want to talk to you more about nutritional ecology and feeding behavior. And I think I'd really like to actually do that with somebody who focuses on feeding behavior in a completely other species to get a nutritional ecology perspective. Maybe we'll pull in one of our comparative friends that works largely in the zoo. I think that would be a really interesting concept to bring to pet nutrition. But we like to kind of end off the podcast with just a couple pieces of advice, I think, from your perspective. So the first thing is if we have a pretty broad yet largely industrial listenership, and if they would like to learn more specifically about whether they should consider in vitro or in vivo controlled in-home experiments, is there a good textbook or a series of review papers, as an example, that you would say, oh, this is a really great place to start understanding digestibility?
Yeah, I think Dr. Maria de Godoy from University of Illinois, she wrote a very nice review of methods to evaluate digestibility together with the colleagues of University of Illinois. So I think that's a very good start. Basic descriptions of in vitro methods that are used, but also in vivo assays that can be used. So I think that's a very nice paper.
Okay. I think that, it's always good. It's good to start with a really good review and then dive deeper where there's interest sets. And if any companies are interested in reaching out to you to talk about research, they can contact you. You can find your email online at Wageningen University and Research. Yes?
Yes, it's available. Yeah.
Great. And thank you so much, Guido. I already, like I said, miss all of you greatly. Very much enjoyed the Netherlands, and I encourage our listeners to follow the group at Wageningen University and Research. They are the best animal nutrition group in the world, so there's a good reason to follow them and learn from them. And thank you again, Guido. I can't wait to see you and talk even more science.
Yeah, thanks for all the compliments, Kate. And we also miss you. And all the best, and looking forward to our collaborations.
Absolutely. All right. Take care.
Take care. Bye-bye.